Hello everyone, and welcome to our first session. My name is Cathal. I'm part of Nucleate UK, where I'm a co-MD in Manchester, and I'm also a PhD student in the University of Manchester, working with AI to help engineer monoterpene synthesis. I would like to welcome you to our first panel session. This is on AI and protein design, and it's done in partnerships with Bits and Bio. And it's running in tandem with our Nucleate Activator program. The approach for this initiative involves hosting virtual panels, mentorship and coaching, in-person networking events, and resource pages to help people get more immersed in the field. The next blocks in our initiative are going to be AI and drug discovery and CRO and automation. But we are going to cover more topics in the coming months. Also, as mentioned, we're going to have some networking events coming up, which will be in the US, Canada, and Europe, and also the UK, right? Depends. So come along if you're able to come, if you're in these cities. Additionally, we have our mentorship program, which involves 30-minute one-on-one sessions with any of the mentors, and you can sign up using this QR code. And you can give a preference for who do you want to talk to, but we're going to allocate slots on the strength of your application. Obviously, all this info will be on our website. So we decided to choose AI and protein design as the first topic, one of the topics that we're going to cover, because it's a rapidly advancing field that leverages artificial intelligence, which is obviously quite hot right now, to create new proteins with desired properties and functions. This area of research is significant for various applications within biotechnology. And one of the key advances in protein designs involves generative modeling, which we'll be covering today with algorithms like RF diffusion and Chrome, which utilize diffusion models to manipulate complex multidimensional data. This allows for the creation of protein sequences predicted to fold to specific structures, which can range from naturally occurring shapes to the de novo designs. The success of these first algorithms first came into the mainstream with the unveiling of AlphaFold 2 at the CASP competition. But AI and protein design is not just about designing new proteins, but also understanding and manipulating existing ones. Companies in this field often start with existing proteins and seek to improve their functionality. Such as efficacy, stability, and reduced toxicity. The integration of AI in this field speeds up the development and testing of new or improved proteins and can greatly reduce the time and cost of improving attributes. So to talk about this topic, we have our amazing panel today. This will last around an hour, and then we'll take some questions from the audience for the remaining time. So for the panel today, we have people who've come from all different backgrounds, focusing on different problems and are at different stages of their research journey. First, we have Manu Ponapatti, who's a Cornell graduate and is currently pursuing a PhD in the MIT Media Lab, specializing in the intersection of machine learning and bioengineering to enhance the biocompatibility of proteins. We have Paul Kim, who previously worked at Bayer and the New York Structural Biology Center as a ML researcher, and is a second-year graduate student at the Baker Lab in the University of Washington, working on deep learning methods for the generation of de novo binding proteins, DNA binding proteins. We also have Alex Tong, who graduated from a PhD from Yale and is now a postdoctoral researcher at Mila in Montreal, and is also a co-founder of Dreamfold, a startup which focuses on generative models for protein design. Last but not least, we have Adam Moyer, who's got his PhD from the University of Washington, where he also worked in the Baker Lab, and he's the co-founder and the director of molecular design at Vilya, where they're using AI to create a novel class of medicines to design new molecular structures not found in nature. So, do you all want to quickly talk about why you got into this field, why you think it's important, and briefly just go into what you're currently working on? I guess starting with Manu.
Okay, yeah, awesome. Thank you for having me. I primarily come from a background of signal processing and electronics. I think I've always been very fascinated with biology. I think primarily because I think it's one of the most like elegant signal processing systems that's out there. I think for at least the way I view AI for protein design is that I think it allows to, kind of enables us to sort of manipulate biological systems. But at the same time, I think it sort of helps us come up with a language to sort of describe and understand how nature designs these kinds of systems. So in that same vein of like trying to model biology as a system and combining that with generative design, most of my current work focuses on sort of building tools that both combine computational immunology with some of these generative pipelines to remove immune T cell epitopes in a de novo design membrane protein.
Maybe I'll go next. Yeah, so thanks for having me for stuff. Yeah, I've sort of worked at from computer science perspective for a while on sort of ML and bio and particularly cell bio, but from sort of a generative modeling perspective. And so I've been working on sort of flow models and models over time for a while. And so when the recent sort of protein design using diffusion and these ideas came out, I was very interested in how sort of you can apply this sort of modeling approaches to this new domain where you can, control things, right? So I think proteins are very nice in many ways compared to cells. So I'm very excited about how you can, build a lot of things easily, right? Sort of cheaper in many ways. So sort of the generative modeling and active learning really interests me. So that's what I'm working on now. And with Dreamfold, we're working on sort of how to design these with the generative models plus with the active learning from sort of large scale assays.
Cool. Paul, do you want to go next?
Yeah, sure. I'm Paul. I started off with more of a chemistry and computer science background, and so my first job was more in small molecule drug discovery with machine learning. This was back in the old days when everyone was trying to use LSTMs and like autoencoders to learn hidden representations of small molecules. And yeah, I got more into protein design because one thing that I was a little disappointed by in the small molecule drug discovery field was often a bit of a disconnect between what's going on in the dry lab and what's going on in the wet lab. And I think this is partly because molecular synthesis is a pretty difficult problem. With proteins, we have the advantage of being able to hijack the protein synthesis machinery that cells have already created for us. And so it's much easier to then generate in the real world arbitrary protein sequences that we've come up computationally. I also think it's really cool that we can, tackle some really difficult problems with protein design, and so that's what drew me to the field. Yeah, so right now I work on adapting the generative models that we use in the lab, RF diffusion, to work for making different types of DNA binding proteins. Yeah, this is what I've been working on for around the past year, and it's been a lot of fun.
Cool. And Adam, do you want to go last?
Yep. Yeah. So, I started out as a chemical engineer. So I have kind of like this chemistry perspective, and kind of like an abiotic perspective to proteins. And I went to David's lab because I was an engineer and I wanted to optimize things. And I thought what was better to optimize than like at the atomic precision. Right? If you want to make something perfect, you got to make it perfect all the way down to the atoms. And so, yeah, that's kind of how I got inspired to the field. And then currently I'm working at Vilya, a startup company where we're designing macrocycles. So we're working on like abiotic molecules that kind of go beyond proteins.
Okay, awesome. By the way, I just wanted to say to the attendees, we're going to have a half an hour Q&A session at the end, and you can ask your questions now, and we will ask the panel at the end of the session. Okay, I think you guys covered it, but does anyone have a part of the field that excites them the most they want to talk about? I think some of you, I think a few of you already said when you were giving your intros, but
This is not my subdomain, but I think enzyme design is really cool, and it's kind of one of the hardest problems right now in protein design, I think. And yeah, but something that we're making, I think, gradual progress towards. So I think that's really exciting.
I'm really excited that sort of machine learning people have another domain that they're doing besides just images and text. So hopefully we'll see more of this.
Yeah, yeah. I remember I was looking for a PhD a few years ago, and it was all just image-based, like ML, like nothing. That was the only biology application, and now the past couple of years, like it's absolutely exploded. Yeah, cool. And I mean, some of you have been in the field. I don't know. Have you guys seen much changes in traditional research due to AI-guided protein design, and do you think that'll change in the future much from how it is now?
I think maybe I can talk about it from a very academic perspective. I think I will say that I still sort of run into labs that I've been trying to collaborate with that are very skeptical of these tools. I think part of the reason is that there is this general feeling that these tools are very good for like making something like a drug, like antibodies or like small molecule binding proteins. But I think a lot of the labs that I work with work on these kinds of hard, like proteins for which testing for function is a really hard thing. I think so then there's a skepticism of doing things the way they've been doing it. But I believe that like slowly, I think as more and more molecules are designed in the lab that are not just like an antibody or like a binding protein, I think you will see a more and more adaptation from experimentalists. At least that's my hope.
So you think it's more to do, going to be more to do with adoption rather than how people use it, like adoption into different areas rather than how they use it in areas?
I think there is both aspects of it. I think there is just at least the people I've collaborated with, there is usually just one person in the lab who really believes in it and champions it. And then that kind of changes how experiments are set up. A quick example, I think some of the proteins I design, one of the issues with it is that the testing for function is extremely hard and expensive. You can test 4 things in a month. And the way people have tested them is that usually they test everything end to end when something, a mutation is designed, sort of, but now they're willing to kind of do a high throughput expression screening. So let's just test for expression first, and then sort of being able to do things in stages and picking things you can do high throughput much ahead of time.
Okay, does anyone else have anything they want to say about that?
Yeah, I think it does make sense that the place where protein design will have the first major impact should be in the biology wet lab, since we're making biological tools. These should be that they're naturally expressed in cells. The like first place that we should try to use them is to make biological experimentation easier. I think one cool example that is being worked on in the lab right now is there are folks designing kind of like scaffolds to put around membrane proteins like GPCRs that will allow them to solubilize in the cytoplasm rather than locate to the plasma membrane. And this makes them easier to study and easier to assay. And so, yeah, there's a lot of tools like that you could imagine building that, might not really have an application outside of the biology wet lab, but I think would have a lot of applications in the wet lab.
I think that one of the there's whole institutes about it, but like I really think like the intersection between like bits and atoms and like thinking about what is synergistic and what does flow all the way from the very beginning when you're designing it on the computer and all the way to the end when you're testing it in the lab is super important. And so it's kind of cool because you kind of have to be like an expert in both domains because there's like an infinite number of potholes going along the whole pipeline. And so you have to be able to like kind of see that and navigate that well. And then the other interesting part is that you kind of have to be an expert in both like engineering and science, where like you have to be capable of like exploring and like designing for like a specific purpose. And so it's a pretty exciting way to work.
Yeah. Alex, did you have any input in that?
Not really. I
This is
think, yeah. So, go on.
Yeah. Well, actually, Manu actually touched on this, and I find it really interesting. So, as we know, a lot of scientists are quite hesitant to, use new tools and use, especially computational ones. Um, have you guys, how have you guys found that in your work? I mean, I don't know how much you collaborate with people who just do wet lab, but I mean, a lot of people nowadays, I think, are very keen to get involved now that there's a big hype around AI and stuff like that. But how do you find that for yourselves?
I think my lab specifically doesn't really have like a big wet lab capacity. And most of the work I do is at the intersection of those two things. So primarily I work mostly with labs that only do wet lab and not a lot of computation. I think I'll see it in two different ways. I think people that I've met who sort of like are championing it are kind of people who are sort of I guess grew up with this, like know about ML and kind of knew like sort of more younger. I don't want to generalize that, but I think more grew up with ML and like understand the new age of like tools and stuff. A lot of people that are hesitant, I feel like, are people who sort of have tried these tools a lot in their careers like previously, and it hasn't really yielded as much results as they think they did. There's a lot of skepticism that they would work. But I think one of the coolest things is the barrier for designing something computationally. There's not a lot of resources you have to invest in it if you're already collaborating with a lab that's computational. So I think a lot of the people I work with are like, okay, let's just try it. Just do a small experiment. Let's try it. If it works, then we can proceed further.
Yeah, so you would say things like lowering the barriers to entry would really help adoption for these things. So stuff like collab fold and stuff like that, where you don't really need it, you just need to input a sequence, really. Yeah.
I also
What
think not all proteins are amenable for design with AI right now. And I think it's important, like for people who are collaborating, to be able to like explain what things you cannot do with the models.
Yeah. Yeah, and that's kind of what Adam said, right? He was saying that there's so many potholes you can hit, and I really like that analogy. So many potholes you can hit along the way, um, that I mean, some of them can go unnoticed if you
Yeah.
don't have the expertise, which is why I think it's so important to have collaboration in the field. What
Yeah.
about everyone else? Yeah.
I think at startups I have an interesting like perspective because everybody has buy-in, right? Like they join the company because like that's our goal, is to use AI in molecular design. Um, and then yeah, what I end up doing more is yeah, explaining kind of like where these limitations are. Um, and anything's like really possible, but some things just take more time to develop and things and more creativity. But yeah, so trying to find like that synergy and that cohesiveness in capabilities in the lab and on the computer, it's kind of interesting because like compared to like image processing or other typical AI fields, right? Like anybody can look at a picture and you can evaluate it very quickly. You can make any image, right? It's just a bunch of numbers. But when you're turning them into molecules, it's a different story.
Mm.
Yeah, at the Baker Lab, we have a pretty wide mix of people who are from purely computational to people who are much more wet lab focused. But I think there is kind of like a minimum level of computer science background that is required. So you don't need to be able to write Python code yourself, but you will need to, for example, understand what the Python code is doing and be able to orchestrate several different Python scripts together in order to run your pipelines and make the necessary changes that you want to make because you ran a pipeline, you looked at it, and you said, oh, I need to change some things. So now I'm going to change some parameters. In general, I do think that, yes, it's true that a lot of the tools have gotten easier to use and more accessible. But if you want to be on the cutting edge here, then having that mixture of computer science skills basically is really important because when the new tools come out, it usually takes around 6 months to a year for those tools to be turned into really, really easy GUIs or like collab fold notebooks. And for the first, 6 months, it's kind of open season. You're just running shell scripts usually. So I think that's, if you want to try to take advantage of that, then the less you need to rely on other people to make these what are essentially research code bases easier to use, the more of an advantage you'll have.
Yeah, I agree with this. I think the sort of, there's definitely a lag between like what is done for the sort of methodological computational sort of stuff, but then like how you apply it to, right? So like the methods are sort of all developed on very easy tasks, right? Easy to do the data collections, very clear metrics, right? So like this as clear as possible, just because, well, that's the easiest to do, right? But then, of course, I think almost everybody experimental is interested in a sort of more interesting problem, right? Like oftentimes they're developed on problems that are basically already solved in some, that's just so you can measure it, right? And so, sort of very helpful to be able to take something designed for, designed to do one thing, but right, adapt it for a different objective, right? And so, sort of the things that we hit on the head with right, with machine learning are very, simple tasks, right? But it's sort of, how do you sort of layer those, right? Going from the simple things to the things that are actually interesting, right? And I think that part is, lots of opportunity there for sort of, people who know both, right? Yeah.
Well, actually, that's actually one of the things I was going to bring up is about the role of interdisciplinary collaboration between like AI experts, wet lab experts, and also people who are on the kind of, they've got experience in both and they're good at that. How do you see the role in like, obviously, basically, I went to this talk recently and they said that AI research, it was about AI and biotech. They said AI researchers end up building better models than a mix of AI and biology people. But right, that is also, well, I don't know. Obviously, as a person who does both myself, I didn't really trust that. But, and I think it's maybe because of what you said, these are kind of set, like they're solving stuff that usually is already solved. And when you're trying to adapt the problem to a different problem, you're going to need that other skills. Would you guys agree with that? Or how do you see the mix or, the individual backgrounds playing into it?
I think I have a controversial opinion at this spot, so I think that's worth sharing. So I was in David's lab from 2015, so I saw protein design pre-AI and post-AI. It's kind of like the two eras, and I'll say I'm upset about AlphaFold in a way because they kind of subverted the problem. They heard like, oh, all these biochemists are saying, oh, we can't do protein folding, and protein folding's so hard. And what the biochemists were actually saying is like arbitrary molecular folding. They were like, oh, look at all the torsion angles, all the combinations. Like these molecules are so complex. And so AlphaFold came in and they solved protein folding, which is what all the biochemists were saying, but it's not what they meant. And I think that subversion is why you see like, oh, AI researchers, like pure AI researchers, do better on these tasks because they're actually subverting like the real problems and not tackling like the most important things. Yes, folding proteins is super important, so I'm not saying that is bad or anything, but they didn't solve the whole problem.
Yeah, I definitely agree. I think the AI is great when you have like a data set and an objective, and pure AI research, right, is great at this, right? Like, I don't think you can beat a bunch of machine learning people just hitting a specific benchmark, right? Of course, all the problems that we're interested in, like outside of language and images, are really not that, right? So once you get beyond that, the claims from AI are sometimes exaggerated, I think.
I think I also don't see building a model as doing protein design. I think I see a difference between those two things. I think because I think when it comes to actually making the protein do something useful in the lab, I think maybe an intersection of those two people will help. But I think as far as model building, obviously, AI scientists, you don't need a biological input, right?
Everyone agree with it? I mean, like with iterations of the model and stuff like that, you don't think there's massive importance in having like a biology background, or?
I think if you're designing something very specific in the lab, I can see, because I think as Adam was mentioning, there's so many other different things that you care about. And I think, and if you talk about pure AI scientists, they might be focused on like one very specific aspect of it. And these are just like, in my opinion, at least just a tiny piece of the pipeline it takes to design something.
Yeah, I think definitely, for actually applying these tools to make proteins that do interesting things, like the actual biology understanding is super important. And there's, I think part of what like is important for labs like the Baker Lab to do is really recruit heavily from like people with MD PhDs or people with more like medicinal backgrounds that where they understand the like disease and the problem better, so that then they can help us like know what types of proteins to design. And I think there are definitely diminishing returns on like how fancy and clever your computational models are. I mean, if I can do like a quick example, so one of the things that RF diffusion does is it's this diffusion model, and so it takes your protein backbone and like moves it all over the place in like a random way, and then the task of the model is to like figure out how to restore the original protein. One of the things that this does then is it breaks up the like natural 1D chain of the amino acids, and this leads to highly non-realistic frames in your diffusion trajectory, because you have two, like an alanine bonded to another alanine, and you would know because they're bonded in the primary sequence that they should be relatively close, but the diffusion model splits them up. So some very clever people had some ideas around harmonic diffusion, where this time you restrict your diffusion process so that residues that are close by stay close together, even though you're noising them, and so even though you're moving them apart. And it's a super interesting idea, but turns out to not really do that much in terms of improving the quality of the designs, because the machine learning model, it's really good at curve fitting, and it just is able to learn the problem of going from full noise to the actual protein structure as it is to this like more constrained task. And so, yeah, I think that there are there are lots of interesting ideas that you can have, but often the thing that you're really bounded by here is like data, and that's where something like increasingly fancy machine learning techniques might not really help.
Okay. Yeah. So actually, that's very interesting. You're saying about how it didn't really help, and they kind of, even though they had this biology expertise that told them one thing. Do you think there's any other? Do you think that the biases of knowing stuff that should happen in biology, how do you think that impacts using AI for like protein design and stuff like that, where you'd make an assumption that is assumed to improve the model because of whatever information that we have in whatever we know about biology? Do you think that this will interfere with like these assumptions interfere with these applications?
No, not really. I think that they can work in parallel, and you can kind of think of it as a like how to best apply your biological priors to improve how you develop your models, and there are cases where that's actually quite helpful. And also, like, then how do you just improve your machine learning model's ability to curve fit, and learn the like joint representations of the data. And so I think, yeah, I mean, a classic example here would be, I think, AlphaFold, where they took this biological prior of, oh, there's this evolutionary couplings information in the MSA that we can use to infer long distance contacts in the protein. And then, but then they also do a lot of interesting triangular attention type stuff that has nothing to do with like really biological priors, but improves the model's ability to learn the task.
Yeah, I think it depends a lot on sort of the data set size, right? Like we've talked a lot about structure, obviously, and in that domain, right, it helps a lot, I think, just because of the limit of data, right? But of course, just in sequence, there's so much more data that, well, you should just throw the most flexible model at it possible, right? And do away with the priors. So yeah, it really depends on the problem. I think especially where the priors are helpful is sort of evaluating the model, right? Like, if you have one target for the model, that's great. But of course, you have many other ways and sort of ideas about, what objectives you should have, right? So inserting objectives is also helpful, I think, for this kind of stuff.
Has anyone else got anything they wanted to add to that? I mean, we can do. I think we should discuss this a bit further in the panel thing, but yeah. So as you said, so I know that Dreamfold pairs with people, companies to generate like large scale data sets for training, because you did mention about smaller data sets and stuff like that. And this this is done. We're hosting this event in tandem with our activator program, so there are going to be a lot of people who will be watching this that will be wanting to know how to overcome this barrier of data, if they're going to train a model or whatever. Do you have any like input in that, Alex?
I think machine learning's been the most helpful, right? Where we have a large amount of data that's sort of noisy and unhelpful for somebody to look at, right? It's like hard to look at, right? If we can make something that's sort of very small and very correct, right? Like I don't think machine learning is very helpful there, right? But the sort of large noisy data sets is where we think the sort of most value will come from these methods. So
yeah, definitely. And I guess will for the rest of you guys, in terms of getting your data and like acquiring your data, making sure it's useful. I mean, do you guys use like data sets that have already been like accumulated, or are you having to generate them yourselves, or?
I think this is like the spot where having that chemistry biology background is incredibly important. Going back to like AlphaFold, right? Like with the MSAs and all of that, like that was like an idea far before AlphaFold. People just didn't apply deep learning to it. And so I think, yeah, cleaning up your data, making sure that your data is right, like in just like that general meaning of right, because the model is going to learn what you show it. So yeah, I think that is incredibly important, and I think everybody should analyze their own training data and make sure it makes sense to them, to their understanding of the problem. And yeah, I don't think you should just generally trust any data that you're training on.
I think I look at data less like, I think obviously data for training AI models, but a lot of the times, I think depending upon the protein that I'm designing, most of the time that the protein that I'm interested in or I'm collaborating on has a lot of work that has been done by people who have done like mutation analysis or like have found different variants of it in nature. And I think a lot of the times I put in a lot of manual hours, like getting all of this data from literature. I think it sort of ends up informing that biological prior that we are talking about. Because if I'm designing a really sensitive protein, I would love to learn from what I shouldn't touch. So I think less about training, but I think less about building those priors. I think it becomes important to have this understanding of like what's important. Yeah.
Yeah. I'm having to do a lot of that literature searching through for my work right now. So yeah, I completely relate. So what do you think? What do you think are like, in terms of good data, right? What kind of things would you be looking for? Obviously, it depends on what you're working on, but what are the kind of key data requirements like for effective training of models for any people. So maybe, I guess, if you're looking at a paper, making sure they've done the right tests and, I guess enough replicates and stuff like that. But do you have any input in that? I mean, if it's kind of an abstract question, if you've not got any specifics, but yeah, just because we were saying good data, and I guess it just applies to the task, right? Um, okay. So what kind of parts of AI in protein design, and any kind of AI approach to working with proteins, like what do you think is the most overlooked part of the space? Um,
I think most AI for protein design is not just a sequence or a structure. I think it fundamentally comes down to being able to predict properties like expression, immunogenicity, thermal stability. I think these kinds of like properties, I feel like, are more important, at least in some of the proteins I work with, for the lab kind of functionality, than any of the structure, like just pure sequence-based methods that I've come into. And that data is extremely hard to come by beyond just one protein family.
I don't know if it would necessarily be overlooked, but I think the world of protein dynamics is something that everyone knows that it's a big issue and is something that we want to know better, but is something that is probably understudied because we don't have the best methods for working with this, especially on the wet lab side. Getting dynamics data is really difficult. I think there are some people working on that field in, especially in cryo-EM, for example, Ellen Zong. But yeah, I think that's kind of like one of the next frontiers that protein design needs to go after.
Yeah, I think to that same concept, like just entropy in general, right? Like what we always model them as like, these static images, and there's a lot of thermodynamics in how they move, what states they can access, and things like that.
Yeah, I think one thing from the sort of AI community is very hard is sort of multimodal things, right? Manu was just talking about how you want to look at literature, right? Well, we have, models that can look at literature, right? And tell you, well, these papers have seen this protein before, right? And sort of, but combining that with a design approach, right, is not going to be done in a sort of main AI, right, conference or research, just because, well, it's very hard to say, there's no benchmark, right? What is, right? How do I design this model to, to do two things at once, right? So these kind of ideas, right, I think are very helpful, but it's very hard to motivate these in the academic research at least. So, yeah.
yeah, I guess that kind of goes on to the next thing. What are the biggest challenges facing the field, in your opinion, obviously?
I think like in general, like real world validation of your models is just it's harder than image generation, right? Like you're not looking at a computer screen and figuring out if it's better. So yeah, I think like that full cycle from the computer to the wet lab.
Yeah, I think I'd agree with that. Also, then the cycle back to, take your wet lab experimental data and use it in some kind of like active learning framework to improve your generation in the future. Getting that like closed loop is something that is something that's it's hard to do. And then I think on the pure design side, making besides dynamics, which is a bit of like an unknown frontier right now, making polar sidechain interactions to like a target molecule is a big challenge. In particular, because like sidechain torsion angles are like hard to predict, hard to design for. There's like Adam said, there's a lot of entropy in that, and that requires then like highly specific design, where you want like a specific atom within like a 1 or 2 angstrom range. That level of precision, I think, is still a big challenge.
Yeah. So I guess that would be, I mean, I was going to ask what kind of things are you anticipating for the future? And would that be one of the things you hope for? Well, I guess you obviously are going to hope for it, but do you anticipate it anytime soon, or do you think it's going to be another while to get this to get down to this level of detail with the models?
I hope that it comes soon. I think one way that it could arrive is through joint sequence structure sampling. That's something that you would have to do because the current framework, the current paradigm is more generate backbones, fit sequences to the backbones, and then test those sequences. And when you're first just designing backbones without addressing the sequence and therefore the sidechain atom positions, you're going to be fundamentally limited in what kind of precision you can get in those sidechains. So yeah, I hope that joint sequence structure sampling will pave the way, but this could also be a kind of a problem where we just don't have enough understanding of the dynamics and the energy involved that causes sidechains to position themselves at certain angles, and yeah, that leads to a lot of challenge. I guess I can go into a little bit of an example here. So at least in the protein DNA binding task, we often want a recognition helix to fit inside the major groove of the DNA, and we in particular want some usually lysine or arginine that's making what we call bidentate contacts with a DNA base. And so these are like two polar regions making a pair of polar interactions to a single DNA base. And part of the problem with this is that lysine and arginine both have a lot of degrees of freedom. And so what we also find is that besides, like in the good binders that we have, what we find is besides this bidentate interaction with the polar with a DNA base, it also requires sort of backing side chains that force the packing of that lysine to fit in the right angle and not like wiggle around and have too many degrees of freedom. I think that level of precision is something that's really hard and not super well understood in this world of protein structure prediction based on crystal structures all the time, and something that we might do better with, especially if we do sort of joint sequence structure sampling.
Yeah, cool. What about everyone else? Is there anything they're really, I mean, apart from high precision kind of modeling, is there anything they're anticipating coming out maybe a bit more in the short term?
I think one thing is like the computers getting better, right? I think that everybody is always excited about, and that's just like a general statement, right? Like bigger models, faster models, more data processing. So we have to keep that in mind that, there's hardware that we're working with. And then I'm also excited about solving the arbitrary molecular folding problem. I think that and interacting small molecules with proteins and DNA and all of those things. So truly solving those problems. Because yeah, once you solve that problem, it's really like open field sci-fi stuff, right? Yeah.
Yeah, super exciting time, right?
Yeah. Just the question, Adam, do you anticipate like using more MD or physics-based approaches here, or ML-based approaches where you collect a bunch of like data on these small molecule conformations, or do you want to do some kind of mix of the two?
Yeah, I think like in data collection, I think having chemical priors is important. So yeah, just thinking about what are the important things that you want to learn, and getting data that represents that to the best of our ability. Yeah.
Any prediction on when you think that might be possible? I mean, you think it's possible with the current data techniques, right? Like sounds like sci-fi to me, but I don't really know enough.
Um, I I mean it acquiring data to test it, right? There's more and more synthetic capabilities that people are developing, and higher throughput syntheses and higher throughput assays for arbitrary molecules. So I don't know when. It depends on how fast you close that cycle, and how many iterations that you have to churn through.
What I've actually started to notice is that a lot of experimental stuff has been like people who are just wet lab have went into stuff that's a lot more easy to generate large and large data sets, right? So like growth assays and stuff like that, which is a lot easier than, I mean, I don't know, whatever, like developing like mutations or whatever like that, right? How do you see that? Do you think this is, I mean, this is just wet lab scientists that don't even do anything with ML, but I mean, I guess they are doing it because people are interested in these data sets. How do you see that changing in the future? Do you guys see people just continuing to do this, or at a certain point, they're just going to start going back to more kind of specific, because more specific low throughput stuff is not when they're relying on like computational tools?
I'm hoping that I think all of these kinds of easy assays that you can run in high throughput, and also the need for like a lot of data for these ML models would also mean like a lot of innovation in like lab automation tools. And so you can sort of in the future, maybe like high throughput stuff could be run with lab automation. I think the biologist's role here is like really fundamentally very important. I think as we start to design more and more proteins beyond just making drugs and like enzymes, I think what the assays is going to start changing. And I think innovation in like how you can sort of test all of these things at high throughput would be really valuable. And also there's, as you said, like some things have to be really specific. Like if you're testing for immunogenicity or like mice-based models, you're not going to do them at high throughput.
Yeah. Has anyone else got any input on that they wanted to say?
I think that no one's going to go back to making small amounts of data. I think one thing, everybody's becoming an AI engineer, right? Or they're trying to figure out how they can become one. And so in this field. And so I think that, yeah, they will figure out how to use the big data that they're generating to solve some question or answer some question that they're trying to answer. And everybody's going to see that having more data is better. So.
Okay.
Yeah.
So you don't think? Oh, sorry.
And I think that there's or to go in a slightly different. I'm just gonna say. I think that there's an interesting trade-off because yes, you want to improve the high throughput nature of your data generation methods, but I think the end goal is that our computational methods get good enough that to actually do design, you don't necessarily need a high throughput approach. In other words, your success rate is high enough that even without needing to even without needing to do something in high throughput, you can get something that works.
Yeah. So, I mean, there has been a lot of talk about the hype cycle in AI, right? Before it actually got involved in biology, I was so involved in biology. So I guess, Adam, you think the hype cycle is gonna, but there is no hype cycle now. We've hit the critical threshold, and it's gonna be AI from here on out.
Oh, yeah. Like, do you think AI can be higher hype? That's what you're asking?
No. I'm more so because with AI there has been a previous hype cycle, right? So,
Yeah.
and now we're going through another one, and it's like AI for everything. Do you think this is going to last now? We've got critical threshold. We've got enough like resources to run these, and we've got data, and in terms of like hardware and stuff to do it. Do you think
I
this
think
is it?
I think the infrastructure's there. I think like the software tools are there for people to learn how to do it. Um, yeah, I guess and I think more and more fraction of papers being published um, like are genuinely using deep learning in a meaningful way. Um, and I think people are seeing the impact of that. Um, yeah. So yeah, I mean, I don't know. I mean, there is one thing that I always say is that like the molecules that I design tomorrow are going to be better, right? Just broadly, right? Like so like the field is just getting better. We design molecules better, and I think people are going to see the impact of that, and it's just moving forward. Um, yeah.
Yeah. Does everyone agree?
I I don't know. I'm less certain about this. Like, I think there's a big limitation, right? Like, if you can make your problem into function fitting, right? I think AI is sort of that's great, right? But there's still a lot of problems where, you know, the understanding, right, the causality you cannot do with a big experiment, right? Like you cannot do with an observational experiment. You know, maybe in all fields we can do large scale interventions, right? But, you know, right, protein design is great in that you can do this, right? But not all fields are like this. So I think there's nothing that can beat, right, a small randomized trial that, you know, tests your specific hypothesis, right? So no machine learning is ever going to get there, I think. So, you know, while the big essays are very useful for sort of generating hypotheses, right? Sort of, you know, right, use GPT to draft your email, right? Like I think for your specific problem, there's always going to be, you know, the sort of the specific knowledge and you know, one specific experiment that will get you sort of the answer that you're looking for, right? Certainly, rather than, you know, maybe it will, yeah, maybe you'll get a guess, but I think we're not even close to machine learning methods that are, you know, going to be certain, right? So, yeah, still a long way to go.
Sure.
I think I'd agree with you, Alex, especially on the no machine learning is going to replace a randomized controlled trial. That's probably that might be really disappointing news to the AI for clinical trials like industry. But yeah, I do think that there is a lot of real value in doing things in the real world.
I I guess I'll just say that I think it'll be better than random, right? So that was another point that had written down here is that like you shouldn't. I don't think you should be doing anything random anymore. Um, I think you should be learning on maybe your first attempt is random, and then after that, like you are learning always, and you're making decisions based off of previous data. So I don't know. I think the age of random is in the past, personally.
Okay. All right. So we're about to get into the Q&A bit, so it's just going to be the last kind of couple of topics. We've already covered like what we think is going to come up in the future and stuff like that. But yeah, is there anything you would like to specific? Like, I mean, you guys have seen each other's bios and what. Is there anything you want to ask the other panelists before we get into the Q&A session? I mean, I can go on another topic while you think about it. I can just quickly go on to another one, but yeah, if you want to ask the other panelists, we can do that just before we start the Q&A from the audience, because apparently there's some really good questions that have come up.
Actually, Alex, I'd love to ask you what the sort of idea, the motivating idea behind Dreamfold is, and whether you guys are focusing much more on structure-based models or sequence models, or you're agnostic and you just want to use what works. What are you guys thinking about?
Yeah, we're currently interested in structure-based models, but I think the sort of so we're very interested in sort of fine-tuning and training, like sort of how do you train generative models knowing something about your right? So if you don't have enough data, right, so we can train a generative model on some data and it's pretty good, but how do you make that, you know, do what you want, right? Like, you know, match specific, right? Like I have this objective that I want my proteins to be more like this, right? So this is sort of our, you know, we think we can make AI sort of guide towards what you want basically, rather than just sort of generate from your data.
And do you imagine kind of like a single unified model with one set of weights to rule them all, or do you think it would be better to fine tune for separate tasks, and or maybe do some kind of like mixture of experts type thing? I don't know how that would work exactly in structure generation, but that does seem to be what works for text.
Yeah. So I think the sort of, if there is a place where you have a lot of the data of the same type, right, then this is the place for big models, right? Of course, that's not the case in most interesting tasks, right? So I think these big models are useful for as a prior, right? But of course, you need to fine tune it to whatever specific thing you want to do. So I think same as language, right? There's going to be a lot of sort of base models, right, which are interesting, but also the development's also going to be in how you tune these your specific tasks and sort of the very low data regime.
That makes sense. Yeah, I think the Baker Lab kind of takes a similar approach right now, and that leads to a lot of pain on the software engineering side, because you have 1,000 different branches from your original like trunk, and then you want to like start to merge ideas together, and it becomes really, really complicated really fast.
Okay, quickly, just before we get into the Q&A, I think, do you guys have any advice for anyone who who isn't in the field at the moment, but is looking to get into it? Obviously, aside from coming to these amazing sessions and looking at the resource list that we've developed, but what other advice would you give them?
I mean, I think some of these tools are fairly easy to try out, right? Like there's a lot of people putting work into, just doing it, right? And so I think like looking at sort of what's generated, right? And just seeing sort of the most, what it looks like, right? Sort of what tasks people are interested in, right? It's sort of the classic problem of, well, I know something about training models, but maybe, I don't know anything about what is the problems with the field, right? And so sort of trying to figure out, well, what people think about these type of models when they run them, right? So there's lots of blogs about, somebody who knows a lot about a specific field trying it and saying, yes, these are still the problems, right? So I think understanding what the problems are still in this space is quite relevant.
Yeah. I have general advice that has to do with AI. I think that if you're not figuring out how to use AI in your daily life to like improve your productivity, it's important. And not only just like figuring out how LLMs can like improve the way you do things, right? But figuring out how you can do things so that LLMs improve how you do it, right? So like don't like try to figure out how to make ChatGPT work for you. Like figure out how you should work for ChatGPT to increase like your total output. I think that's like just a framework shift.
Yeah, and as Alex said, these things are becoming more accessible. So like obviously ChatGPT, but also like other areas are becoming more accessible. So just trying it out, I guess, is what you guys are saying, and then you'll learn how to adapt yourself to it.
Yeah.
I think in terms of advice, I'd say, and really good advice on like getting good with like ChatGPT. But I think because this field is so big and so interdisciplinary, I think it's important to try to find your niche, and then like if you're just trying to get into the field, pick a niche and then really like study the things that you need to become an expert in that niche. From where I'm sitting, I think I kind of see like three main places where you can sit. One is like much more machine learning understanding. And so they're like people with like a pure ML background who then learn a little bit of the biology here and there, enough to kind of get the context, but don't really focus on a specific biological problem, and are more on the like pure algorithms development side. And then there's kind of on the continuum of like pure ML to pure bio. I think there's a space for people who are kind of a little bit in between, where they have a little more bio background, maybe a little less ML background, and are able to then tailor the ML models to the specific use case of the biological problem. And then I think that After that, the best place to sit is actually someone with much more biology understanding and like doesn't worry too much about how the machine learning model is developed under the hood, but knows how to use it, knows how to orchestrate it with other like machine learning models, and can run computational pipelines on their own, but really has a biological problem in mind that they're trying to target, that they're going to try to do with currently existing tools. So I would say like those are the kind of three places. There might be more, but at least from my perspective, those are the places where those are the kind of the niches in the field right now where you can make the most like differential impact.
Yeah, and I think make friends all along that spectrum. So because yeah, one person, all of them can't work independent, really, but it's true.
Have you anything you wanted to add, Manu?
I think like just echoing everyone's points, I also feel like when I first started in the field, I think coming from someone who has not taken biology since high school, I found that like, oh, I have to learn like all of it before I start jumping in and like writing models. I think the barrier for entry for any AI engineers trying to get into bio is actually pretty low. I think if you focus on a specific niche, as like Paul was talking about, I think it seems scary, but I think it's something you'll learn along the way. And you may never learn everything that you need to learn about biology or chemistry, but I think you can still make valuable contributions as an AI engineer. And I think there's a lot of resources, like there's seminar series that people do. I think generally I found this community really like friendly and like open to collaborations for any biologist. I think like there's a general, everyone wants to solve these problems and the community is very collaborative. I think hopefully you'll find someone who's working in the field. I think that would be a great introduction.
Yeah, definitely. Making friends along the way is a good shout. Yeah, okay, right. We're going to go on to the Q&A section now. So I have one here, which is actually quite interesting. What do you think is the most misunderstood about the field from the general public and non-experts and scientists and investors?
I think a lot of people believe AlphaFold solved the thing, right? Which I think, yes, I think everyone can agree here. It's definitely not true.
Yeah. I think that's what I was.
Sorry.
That's okay, Manu?
I also think like in my opinion, at least, there is an overestimation of what AI can do right now. I think there has to be, I think it is still an extremely resource intensive thing, even if you're doing AI for protein design. I think high throughput experiments cost a lot of money. So does like AI models, like even running inference costs a lot of money.
Yeah. Okay, what about this one? What is the availability for of data for protein design beyond AlphaFold? Are there any other standardized data sources? I guess they're talking about like PDB and stuff like that. But are there any other standardized public data sources, or is data largely generated as needed in lab?
I mean, I guess on the sequence side, we have tons of data over at Uniprot, Uniref. Unfortunately, I think more like specific functional data or like, better annotated data than just the structure and structural annotations in the PDB kind of is a Wild West. Like there are random like data sets being curated by professors and labs kind of privately, or not privately, but like, just a small operation, not really working to integrate with other data sets that much. Yeah, and I think that making that nicer would be a big help. But it's hard to kind of coordinate incentives to make something like that happen, especially because it would then just be publicly available data.
Yeah, as Manu said earlier, often, I mean, if it is already publicly available but it's not annotated, it takes a lot of like sifting through lots of literature, and so yeah, making them more publicly available, and I guess the incentives is definitely something that we could use in the future. All right. So this one says, why are GANs almost abandoned an ANN generative approach in bio. GANs are very far from reaching their full potential, and it looks like fashion shapes AI research direction. Um, uh, uh, isn't it a bit counterproductive? So I guess why are GANs overlooked?
I think generally AI just tries to call things differently, right? Like, most of the diffusion models, right? They all have a GAN component, right? So the latent diffusion models all have a, GAN in them, right? Like adversarial training is not going away ever, right? So it's the ideas are still there, but sort of what you write around is different, right? So what's emphasized, sort of we know how to approximately train an adversarial model, right? We sort of push the limits of yes, you can train it. I think in just practice, right? Like it's harder to make a GAN train well than a diffusion model, just from a purely like playing around, playing with hyperparameters, right? GANs are just much more sensitive to weird things, and so I think that's the biggest reason why they went away. It's sort of it's less, yeah, it's harder to train them. So if you can just make a diffusion model, it's easier from an engineering perspective to make it work well. But there's still places for adversarial training. Even in the state-of-the-art models that say they're around diffusion, but really other things are still very helpful.
Discuss anything a little more. Specific question to Paul here. How does he think transformer ML architecture will contribute to enzyme design specifically, or any other specific ML algos that seem more promising to him?
I think for enzymes, actually, the question is less in the specific ML algorithm to be used. I'm sure it will end up being transformers because transformers work well. But the real question is actually, I think, more on the like quantum mechanics side. How do you model the transition states that you want your enzyme to be able to achieve or produce? That's something that is not really a machine learning problem as much as it is a physics and quantum mechanics problem. But I think that's kind of the first step to, being able to design a protein then is to know at least within the active site, like what is the chemistry that you want to happen. And therefore what is the geometry and the energy states that the side chains must be able to accomplish in order to make your enzyme active. Yeah.
And then I think another interesting concept here is making sure that you choose an enzyme that an enzyme paradigm that works with deep learning and the resolution that we have. Right. There's plenty of enzymes that, we'd have to have like vibrational bond mode precision, which is not there for models and will be a while probably.
Yeah, I think there's definitely a room for combining like deep learning based approaches, de novo based approaches with then directed evolution on top of it, because like you say, like that level of precision is so hard to achieve. And for a lot of these directed evolution problems, you need some kind of starting point. Maybe you can use de novo design to get you like a really crappy enzyme for your task, but then you can optimize it with directed evolution to make it actually useful.
Yeah, and I think as well that kind of speaks to getting involved with these tools just even at a lower point, because then you're able to daisy chain together these things, these approaches. Okay, question. As someone interested in and studying physics, I'm curious what roles you guys see physics playing in the future of protein engineering. Are physical-based methods going to be fully replaced with AI? I mean, I guess that's just talking about molecular dynamics and stuff like that. But yeah, Manu, did you want to say something?
No, I don't know if they will. I think maybe we will see more and more models that sort of like I guess will incorporate information from these physics-based models, because I see a lot of models that, for example, testnet groups at MIT kind of work on, like in trying to build sort of more physically valid operations on top of these atomic scale things. Maybe we'll see a lot more of those models. And I think even in the last like sort of set of conferences, we've seen a lot of people trying to approximate what molecular dynamics trajectories sort of look like, whether it's for one specific family or not. I think we might see those kinds of models. I am not sure if that will fully replace all kinds of physics-based modeling we still do, because at least I, to this day, still rely on doing MD at the end of everything I do when the protein I'm testing is very expensive. So I still don't see that being replaced anytime soon.
Yeah, I guess one of the questions is like how many models have like Leonard Jones in them, right? And I don't know how many. I don't know an ablation study where someone like made a model with and without Leonard Jones and saw how much better it was. But
Actually, in our diffusion models, we messed around with like a Leonard Jones loss, and it didn't really improve anything. So
sure.
There's one data point for you.
Yeah.
Okay. And I think as well, do you guys, from what Manu said, I think the kind of thought was that there would be increased collaboration and use of physics-based approaches if you for this AI and biotech, rather than the physicists who are getting involved will be, or the physics things will be obsolete in the future.
Sorry, what was the question exactly?
Oh no, I was just trying to saying like because Manu was saying that. I guess what I was just what was the last question? I forgot, to be honest. Yeah, are physical based methods going to be fully replaced with AI? So yeah, the idea was that there would be more. There would be an increased role of physics playing a role of protein engineering rather than a decreased role, as we've got all the data from them and we're never going to see them again. I guess I was just touching on that.
I think one way that physics-based approaches could, like MD, for example, could start to like be more useful than just pure ML or ML with like physics priors is if more, like I was talking about, more like QM stuff gets incorporated into these physics models. Unfortunately, like, QM computations are so expensive. And so you either are left with like you working with a really tiny system. And if you want to make your system any bigger, then you need to start making really strong approximations of your QM, and you head more into just like balls on springs approach of MD. I think if we can like make run like a larger scale MD level simulation, but with QM precision, that would be the thing that makes physics-based models now like a lot more useful than ML. Hopefully then ML would start to, use that the data coming out of these models and then improve as well. But yeah, I think that's what I'd hope to see in like developments on the physics side.
Cool. Okay, we've got a question here about what are your thoughts on recent projects like Future House's AI scientists and the open dataset from initiative from Align to Innovate? And where do you see these projects in 1 to 2 years from now?
Well, I talked to the Future House people at NeurIPS, and I was very impressed about how you can do literature searches and this, right? I think hopefully they'll be able to integrate it more, right, into sort of how you actually write other types of models and sort of predictions for other types of tasks. So, that's where I hope it goes, right? The sort of multimodal idea, right? Using the language models within our sort of other types of generative algorithms, I think will be very exciting if this is possible.
Yeah, I've kind of decided to not have strong takes on like LLM future development stuff because I was totally wrong about what you could do with just like a masked character, masked token prediction task. I always thought that you needed to have grounding. You know, it's like, of course, that's how humans learn. They like pair the image with the word, and like there's this semantic understanding of the real world that's necessary. And turns out you don't need that at all. So having got that one wrong in the past, I don't think that I don't want to make any claims.
Yeah, fair enough. Okay. Well, this one is kind of, well, I guess is has slightly been answered by Alex and Adam in just what they do. But if you would start an AI uh, for drug development company that involves LLMs, agents, and tool use, what would it be? I mean, we've got two co-founders in the chat that are working with AI for in biology, but what specific, I guess, would be the most important, I guess.
And then I guess that it specifically was with LLMs.
Oh yeah, LLMs, agents, and tool use.
Yeah, I haven't thought a lot about that. I'm trying to figure out what tool, like how I can use tools, right? So I think there's like a demand in that, right? But yeah, I don't know. It seems like a B2B type of proposition, not there's definitely no like general consumer that's looking for that solution, I think. So
I mean, I guess so the thing that AWS did was like abstract away the like complexity of the computational like server side of doing like a internet or like uh, computational company. I think that would be I know that there's like a lot of barriers to this, but it must be that in the long run that also happens for the wet lab biology side. Like um, once you can get the experimentation like good enough and flexible enough and accurate enough, um, there like the ideal case I think is that all of our wet lab experiments are done via an API where we like write our instructions as like commands in code and they get executed like the same way every single time by robots or like by some level some type of automation. I don't imagine like fully articulated robots in movies, but more like a bunch of linked microfluidics devices and um, like automated pipetting machines and stuff like that. And maybe that would require this like specific combination of LLMs, agents, and tooling. Yeah, I think this is obviously a bit of a pipe dream. Emerald Cloud Labs is trying to do this, I know, but there's like a lot of challenges here, and there's so much like uh, messiness in wet lab like work where sometimes you have to like remember to shake the thing before you put it into the machine, and it's like that's such a that's not hard for a robot to be able to do. Also, like hard to sometimes remember to input that in your protocol. Like there's tons of those types of weird finicky things about the wet lab that make it currently much easier for humans to do. But from like a pure like economic perspective, it must be that we then like reduce the cost of doing wet lab experimentation by like putting it in some kind of shared cloud system rather than each individual company needing to like buy their own wet lab or like contract out to some CRO. Yeah, I think that's got to be the future.
yeah, there's definitely value to extract there, considering that like the what you're generating then for your customer would be very valuable to them, right? Which is the experimental data. So
Yeah, I think as well the consistency is a big point as well. With the enzymes that I work with at the moment, the consistency of results from literature, they can end up producing something completely different. And it's to do with these kind of niche bits that humans do slightly differently. I mean, I guess as well, like with crystallization, it's a big thing. Like, I mean, there's a lot of like magic involved, basically. That if there was cloud labs more available, or automation stuff like that, then these things will be a lot more consistent, right? And we're actually going to cover CRO and automation in one of the next blocks of our initiative. Okay. Can you describe unavailable robotic equipment that you think would be most important to validating the work done with AI? Hmm.
I don't know. I guess one thing would be getting crystals instantly. Um, I don't. Yeah,
Yeah. I
proving
mean,
that.
that's kind of a pipe dream, though, like, right?
Sure. Yeah. Yeah. I don't. I guess I don't have like anything. I think there's a lot of problems to solve, right? So I don't know where you draw the line on like which one you should or shouldn't do, but
Actually, for well, I mean, Adam, I guess brought it up first because what can you talk more about how we should work for ChatGPT and examples? If you've anything more to add there. I mean,
uh,
obviously, this is open to everyone as well.
yeah, I kind of had that one liner, you know.
Well, it was a great one-liner. I mean, just I think as well, like just testing them out, and you will like what you said was like by testing them out, and when you use them, you'll get more used to using them, and then you'll work out how to work yourself so that they can benefit you more. I
Yeah,
think was what you said, but I
yeah.
think there's not much to say rather than just giving it a go, right?
I guess like a specific example is like I write my code in such a way. I write my code the way that Copilot auto completes. Um, I guess is like the fundamental thing. So, but it's ended up like changing the way I structure what I write and things, and yeah.
Yeah. Right.
I put my ChatGPT custom prompt in the chat also.
Oh,
Yeah, this is stuff that I've found on Twitter and stuff like places that it really helps the output because the current models are very like wishy-washy and hedgy and annoying to work with sometimes. They're always like, "Oh, this is a very unique thing," and yeah.
yeah, awesome. Thank you. I'm gonna download that and have a look at it later. um, what are the what are those checks that you need to perform to trust the data? What gives you the most confidence? Metadata, orthogonal measurements, comparisons to well-characterized results.
Yeah, I think you think to yourself, what must be true for this to be good data? And then you go and look at that specific thing. And those things should be like very detailed and like one like subcomponent of a subcomponent. And then you like go through and check various little small details and see like what thing doesn't make sense. Um,
Okay, right. I think we've answered quite a lot of these questions. So trying to find out one we haven't covered. Um, um, big discussions in AI is happening around the roles of broad foundation models versus many specific models finely tuned around some specific domain space. How do you think protein structure function models will fit into that debate?
I guess I can say that like I always would prefer to work where like something's been pre-trained. I don't want to train everything up from scratch every time. And I also think that it works better to have pre-training. So I imagine like systems of AI models is better.
Okay, so this one says, for protein applications with end use in humans slash animals, I agree with Manu. Immunogenicity is an overlooked slash non-talked about issue. I would love to hear more about AI approaches on how to avoid B and T cell responses if at all possible. If anyone knows about this and has anything been done with the space, or what are the roadblocks? If anyone, um,
Maybe I can answer that question. I feel like I think for better or for worse, I think trying to remove T cell epitopes or any kind of immune response, like from the things you're designing, is an extremely hard problem to achieve. I think if we could do it, I think it's a very scary future. I think the way I've seen it is sort of like a lot of the times I feel like the research of like trying to reduce immunogenicity of something, I've seen more of it in like a pharma company or sort of like in some of the newer like AI-based like startups. I think less so in the academic world. I think most of the focus in the academic world, at least, is in trying to study what kind of like ligands get expressed on the cell surface. We're studying more about like understanding immune systems and immunology. There's a lot less of people trying to make something like non-immunogenic in the lab, at least, mostly because it's extremely expensive to clinically validate. At least in the work that I do, I'm basically trying to sort of use sort of like if I have this protein, like and kind of seeing if this protein is very much designable, can I push like in making a version of it that at least with what data I have about immunology is, predicted to be non-immunogenic, but it should work in the lab. I think in my case, the protein is extremely sensitive. So my hypothesis is that with all these filters, if it does still function in the lab, there's a greater chance that it might work in the clinic. But I think it's extremely hard for any academic lab to get that pipeline like working. Because also I think there's a hypothesis that most mice models may not even be the accurate representations of our immune system. So even if you say that this worked in a mice, that you're never going to test that clinically.
yeah. Okay, awesome. We are coming up to the end here. If any of you want to have anything, have you got anything else to say? Is there anything that we covered that you wanted to bring up quickly before we finish?
Vilya's hiring deep learning scientists to work on molecular design. So
Awesome. Anything else? Okay. Right. So that was brilliant. That was super. It went super. I don't know, easy for me. You guys were very chatty, which is great. Thank you so much for the panelists to come along to cover this exciting field. As I said before, we have a number of similar initiatives we are putting together as part of the AI in protein design block of the AI in Biotech initiative, including a resource page and an introductory overview session with Paul, where he goes over more in detail the kind of background of AI for protein design. You can follow along with our AI and Biotech initiative, where we'll be covering other areas, including drug discovery, CRO and automation, data management and regulatory compliance, as well as biomedical AI, ethics and alignment. There should be a mentor sign-up link in chat now, as well as a survey link discussing this event. But if not, it will be emailed out to you later. And finally, I would like to thank the Nucleate team who helped organize this, and as well as Bits and Bio, who were partners with this for this initiative. And finally, I'd like to thank the panelists again, and thank you all for coming along today.
thank you.
Have a nice day, guys.
Thank you. It was a lot of fun.